Transcription and understanding how genes get turned on and off really underlies every biological process. The brain is a very complicated organ. It's our most complicated one, and understanding how genes get turned on and off are really important for not just normal human biology, how our brains work, etc., but also for pathology, for disease.
Despite every cell in the body having the same genetic code, different cell types need to express different genes. Transcription factors work as a mechanism to make sure that each cell type expresses the type of genes that it needs to express in the right time, in the right place.
What's important for all the cells in our bodies is that you make particular proteins at certain amounts, and it's different for different cell types. So it's a really highly orchestrated system. We care a lot about this because we do know that much of disease of all organ systems, but particularly in the brain, are ones that if you have a perturbation of that process, even at one gene, it can lead to a really serious disease. So some individual genes can cause Alzheimer's, they can cause Parkinson's disease, ALS. One single gene causes Huntington's disease when there's aberration in that expression of that gene. So we study what those proteins do in the brain, but also what controls the expression of that gene and what goes wrong when you're making too much or not enough of the protein.
Early studies looked at homogeneous cell cultures grown in a dish, but what we wanted to do was better understand human disease, so we studied transcription factors in human tissues. One of the complications with that is that each tissue in your body is made up of different cell types. Those different cell types might give different signals and give noise in our assays. So one of the approaches that we take to look at individual cells is to use an approach known as flow cytometry, where we can label cells fluorescently, and different cell types can be separated using a series of lasers and filters and electric fields. And it allows us to separate thousands of cells per second, and once we have those different cell types, we can do individual assays on them. We're also starting to use new innovations in cell labeling, where we can actually barcode individual cells before doing the assays, and then once our experiments are done, we can link the results to those original barcodes, and it allows us to assay millions of cells and get very granular with the cell types and see what individual cell types are doing in the individual molecular pathways that we're testing. By studying transcription factors in specific cell types from human donors, we were able to link the activity of specific transcription factors to psychiatric disease and in basic human health.